Welcome, everyone. Thank you to those who are joining right now. Good afternoon to you all. Thank you so much for joining us today. My name is Brooke, and I'm delighted to welcome you to the Annovis Patients webinar. Today, we will answer questions that we received from patients and their families about our clinical trials. Before we begin, I would like to remind everyone that today's call may include forward-looking statements. Perhaps you can pass to the next screen and see these statements that are based on current expectations and assumptions and are subject to risks and uncertainties that could cause actual results to differ materially from those projected. For more information on the risks and uncertainties related to Annovis Bio's business, please refer to the forward-looking statements and the company's latest SEC filings. To guide us through today's presentation, I am pleased to introduce our speaker, Dr. Maria Maccecchini, the founder, president, and CEO of Annovis Bio. We have received many thoughtful questions via email ahead of today's webcast that will be addressed after a brief presentation. Additionally, we will have a new feature today. There will be a live Q&A section at the end where anyone will be welcome to raise their hand using the raise hand button on the taskbar, and you'll be invited to unmute your microphone and pose your question live if you choose to do so. First, we will pay attention to Dr. Maria Maccecchini. I'll turn it over to you.
Thank you so much, Brooke. So as Brooke just said, I'm going to give a quick presentation, and then we'll have the question and answers that you sent in, and then we have the open mic. So as for the last 10 years, we have known that Alzheimer's disease is more than just plaque. And in 2018, Peterson from the Mayo Clinic actually told us that Alzheimer's disease was plaques, tangles, Lewy bodies that are usually associated with Parkinson's, TDP-43 aggregates, that's a new toxic protein, vascular dementia, and other. Interestingly enough, our drug is following exactly what the brain has been doing. Our drug inhibits the overexpression, the high levels of toxic proteins, be they amyloid, be they tau, be they alpha-synuclein, or TDP-43. And by inhibiting more than one neurotoxic proteins, we were assuming, maybe 5 or 6 years ago, that it would work better than if we just inhibited one neurotoxic protein. So we did over 10 clinical studies, and I'm just showing the last 2, which in this case is a phase 2/3 Alzheimer's study, where it did not work in the total ITT population, but in the mild Alzheimer patients, we see a beautiful dose response curve that is highly statistically significant. Because whenever you look at the subgroup analysis, the question is, was it cherry picking? We actually looked at this subgroup analysis in great detail. And what you're seeing here is the patients, the different types of patients in which the drug showed an effect in the ADAS-Cog. So on top, you see overall, that's the data I just showed you in the graph. Then you see that age doesn't make a difference. Apolipoprotein 4 carriers or non-carriers do not make a difference. You may know from Leqembi and Kisunla that ApoE4 carriers do not respond as well to these 2 disease-modifying drugs, and they show a lot more side effects. In our case, we don't see that. It works in carriers and in non-carriers. It works in heavy people and in light people. It works in Hispanics, Latino, and non-Hispanics or Latino. It works in women. It works in men. It works on top of analeptics, which would be Aricept and Namenda. It works on top of antidepressants. In fact, it works very well on top of antidepressants, and it works on patients that don't take any of these drugs. And then finally, race, black or white. We don't have any black placebo patients, so we couldn't calculate the forest plot. But this shows very nicely that our drug works in everybody in the mild Alzheimer population. And then we looked at biomarkers, because biomarkers give you an idea of what happens in the brain. And we saw that GFAP, which is an inflammatory factor, is reduced, so there is less inflammation. We saw that neurofilament light, which is a marker of nerve cell health, is reduced. So again, the nerve cells are healthier. TDP-43, one of our 4 neurotoxic proteins, is lowered, and tau is lowered. I want to point out that while I'm only showing you the Alzheimer data of the phase 2-3, we see the same reduction in GFAP, NFL, tau, and TDP-43 in our phase 2A studies, the Alzheimer and the Parkinson's. And this shows how our drug stacks up if we compare it with what is on the market. The yellow line is the natural cognitive impairment increase if patients are on no drug or on placebo. The red line is Aricept. At 3 months, Aricept shows a 1.5-point improvement. At 6 months, it's normal, it's back to normal. And at 9 months, it kind of merges with the line of the patients that were not treated. The blue line is Kisunla and Leqembi. These 2 drugs are disease modifying. As you can see, they do not improve cognition. However, what they do slow the decay. In fact, they're 30% better than placebo. The top line is our drug. The first part is what we know. Our drug today looks 3 times better than Aricept as a symptomatic drug. The dotted line really tells us what we don't know. That is the study we are doing. We want to know what happens after 3 months. Does the efficacy stay up? Does it go down? Does it go down to, and gets down to the yellow line? So it's a good symptomatic drug, but not a disease-modifying drug. Or does it stay up? Can we actually show efficacy through 18 months? Then we know it's also disease-modifying. And this is the study we discussed with the FDA, and we started 2 weeks ago. So we started an 18-month study, where after 6 months, we open the blind and we analyze the data to show if we have symptomatic effects. And we continue the study. The patients will remain blinded. We continue this study for an additional 12 months. And that data then will tell us, is this drug disease modifying or not? So this slide shows what we have agreed with the FDA, that we do this 18-month study with 2 readouts, one at 6 months, one at 18 months. The first 6 months readout we expect to be in summer of next year. The 18-month readout we expect to be in summer of 27. And now we come to Parkinson's. And a lot of you are very interested in why we are not moving forward with Parkinson's, and we are moving forward with Parkinson's. So let me give you a little background. In the ITT population, we saw cognitive improvement. In fact, on the left, you see that in the whole 523 patients that we treated, the placebo gets worse. The 2 doses actually remain the same. This could be an indication of prevention. It prevents cognitive decline. It's only a 6-month study, so we don't know what happens at 18 months. But if this trend remains, it is a preventative. It prevents cognitive decline. If we take the patients that actually are already cognitively impaired and have an MMSE of 20 to 26, we see a beautiful improvement, dose-dependent improvement in their cognition. And again, we did a forest plot. This is a forest plot of the patients with cognitive impairment. And as you can see, it doesn't just work in improving cognition, it also works in improving Parkinson's movement and postural issues. In UPDRS 1, 2, 3, 4, we actually show an improvement. In CGIS, we show an improvement. And then below, we also see improvements, but the improvement is the other way. So the WAIS test, which is a coding test, and the PGIC, which is a physician impression test, they also improve. So again, in the subgroup of MMSE 20 to 26, we see that their Parkinson's symptoms get better and their cognition gets better. And this is the data we have for the total Parkinson's population. The ITT population had too much noise, too much placebo noise, and we did not see an improvement. However, when we did the per protocol population, we saw an improvement in UPDRS 2, 3, 4 total, and again, MMSE. The two graphs I'm not showing you, but it will come up in the discussion, we will also show an improvement in PIGD and in more than 3 years, less than 10 years. So we actually have 3 subgroups where we saw an improvement in MDS UPDRS, and we'll discuss later what we're doing with those 3 subgroups. So these are the milestones for the next 2-3 years, okay? The pivotal 18 months Alzheimer's study has started. We are recruiting 750 patients, and next summer we will have the readout for symptomatic. We will continue the same 750 patients for an additional 12 months, and in summer of 27, we will have the readout for disease modifying. Then what we are doing right now, we have been working on the open label study. We did submit the protocol for the open label study for Alzheimer's and for Parkinson's disease, for both, to the FDA, and we are expecting their reply. Right now, the open label study for both indications is planned to be a 3-year study. And then at the bottom, you see Parkinson's. And again, we are preparing to go to the FDA, and the arrow is blank, partially because of a mistake, and partially because we are not 100% sure yet how to exactly structure the study. But I'm perfectly happy to discuss with you what we are thinking. And I think this is kind of, oh no, sorry, I forgot. So the reason it was blank is we are not 100% sure, but how do we get the money to do all this? And as you see on the pivotal study on top, we have enough money to make it to the first NDA, the first 6 months readout. The additional 12 months is contingent upon additional funding. We have FDA approval to do that. Now, if you look at the FDA response for open label, we don't have it yet, we expect it. So the open label is contingent upon when we get the FDA response, what they want us to do, and on funding. And then the PD study is contingent upon us finalizing what we want to do, and again on FDA, submitting that to the FDA, and again on funding. And this is our whole program for the next 3 years. And I think this is it. So we are up for questions and answers.
All right. Thank you so much, Dr. Maccecchini, for that update on the company's progress. Okay, we have someone raising their hand already. However, just hold that question. I think we'll answer that at the end after we have a few questions that have already come in advance. Is that right, Maria?
Yes, we will answer the pre-questions first, and then what's left, I'll be open to answer anything.
All right. Thank you so much for being ready to participate. Just hold that question a few more minutes. So I will read each question, and Maria will provide the answers. And at the end, of course, we'll have that live open section. So the question 1 is, when will AD phase 3 results be known to you and when to the public?
So I was saying that we expect to have the results or finish the 6-month study next summer. So give a few months for the results to be analyzed, and they will be made public 4 days after I receive the results. The SEC has a rule that you have to report important information within 4 days. So I will be the last person to know, but 4 days later, everybody will know.
Wonderful. Next question, question 2. When will the open label start?
So as I was saying, we submitted the protocol to the FDA and we are expecting them to answer. So I assume we'll have an answer in a few months and then we'll start the study. We are actually working towards starting the study. We're also working towards finding the money to start the study.
Very good. Next question. Will the AD and PD open label use the same sites that participated in the phase 2-3 for AD and phase 3 for PD?
Yes, we very much expect to use the same sites. Now, maybe not all sites will be available. So what we will do, we will actually put all the sites on a map of the United States and then maybe add a few sites where we are sparse. Yes, but everybody will have access.
Okay. Question 4. As it is an open label and you will have real-time data, how do you plan to use and leverage this data?
So from a regulatory point of view, the only thing you can do with an open label study is look at safety. So from an FDA point of view, we'll look at safety. Having 3 years of safety will be absolutely fantastic. It's not necessary, but it will be fantastic. Now, from our point of view, of course, we would like to see if people that were in a study, how have they progressed since they stopped the other study and come into this study? Is that progression different from people that were out of a study for a year versus 6 months? And then how is progression? But that is for us to know how the drug works. The FDA will not put any weight on that.
Okay. Next question. Are there any PD KOL events planned?
Yes, we had 3 because, as I was telling you, we have a lot of data on PD and we're still a little bit scratching our head. We had one on Parkinson's, straight for Parkinson's, one on Parkinson's disease dementia, and one on Lewy body dementia. So we have a pretty clear understanding how to proceed with dementia, whether that be Lewy body or a Parkinson's. We are still Okay, so what came out of these KOLs is that since we have a little problem identifying our patients, we should do a lot more biomarkers. And that is exactly what we're doing. I showed you the biomarkers for Alzheimer's. They're very solid. In Parkinson's, we have a lot less plasma biomarkers that are solid. But we can do a lot of biomarkers, and that's exactly what we're doing to identify the patients better.
Okay, so that takes us to the next question. Do you plan to use alpha-synuclein biomarker as a potential tool to identify correct patients for your PD trial?
Yes, in fact, we will do exactly what we should have done last time, and that is take CSF and do the alpha-synuclein test in CSF. So I do want to warn everybody who comes into the open label trial that they will have to give CSF.
Okay. Next question. With the excellent results that have been achieved by the phase 3 trial for PD, why is there such a long period of time before an NDA is filed?
So as I was saying, we have 4 subgroups in PD. One is cognition, but that we also have ITT. So we have statistical significance in ITT and in the patients that are cognitively impaired. That's a clean group. The other 3 groups, one is per protocol, which is a little, I mean, you say, well, the patient didn't take all the pills. The patient was worse and on than off, the patient, whatever. It's a little shady. I mean, no, it's not shady. It's hard to define exactly what per protocol is because you don't know if the patient made a mistake, if the doctor made a mistake, if they wrote it down incorrectly. The other one is more than 3 years, less than 10 years. Again, we did that to try to eliminate patients that didn't have Parkinson's disease. But that also eliminates Parkinson's patients that have very early Parkinson's disease. And then the third one is postural instability. That's a solid group, but it's a very, very tiny group. And we can't really do a study with a very tiny group. So we have, as I said, we had 3 KOL meetings. And I think the consensus really was to try to measure as many biomarkers as possible. And whereas even for cognition, so that we can differentiate the patients that have cognitive decline from the patients that don't, not just by MMSE, but also by biomarkers.
Okay, thank you. Next question, how successful has buntanetap been as a disease-modifying therapy, and how is this success measured?
So in animals, I know we have data that it works up to 9 months. So in animals, it's definitely disease modifying. A mouse lives 1.5 years. So if 2 years maybe. So if you have efficacy for 9 months, that's an old mouse. In humans, we only have 3 months and 6 months. 3 months in Alzheimer's and 6 months in Parkinson's. That is too early to know whether we do or do not have a disease-modifying effect. In both cases, we know we have symptomatic effect because we improve cognition for 3 months in Alzheimer's, and we improve cognition and movement in Parkinson's for 6 months. So we need to do 18-month studies. And that's what we're doing in Alzheimer's to show that it may be disease modifying. And in Parkinson's, we plan to do the same thing when we get there.
All right. We have a few more of these prepared questions that have come in. The next one is, can you remind us about why you were interested in exploring the relationship between buntanetap and GLP-1 agonists like Trulicity and products like Viagra?
Yes. If you think of health, okay, you need good diet. That's good sugar metabolism. The brain needs just the right amount of sugar, not too much, not too little. Well, GLP-1 agonists do that. Now, insulin does that too. Metformin may do that too. But, okay, GLP-1 right now is a really good molecule to regulate sugar metabolism. So you need good diet. You need exercise. You need to move. Well, that is circulation. Hypertensive drugs actually improve circulation. Viagra is a hypertensive drug. Now, why Trulicity and Viagra and not anything else? I looked at drugs that enter the brain, and Trulicity is the best blood-brain barrier permeable drug of the GLP-1 family, and Viagra is the best blood-brain permeable drug of the ED family. So you have good circulation, good sugar metabolism, and our drug protects nerve cells from dying. So the idea is that if you have all three, that if you have all three, you should be better. And that is exactly what we see in our mice. Our mice do a lot better on the combination than on one single drug.
Okay, thank you. Does Annovis rely on any federal government funding?
Yes, we do. We have written grants.
Perfect. All right, another question. In one of the latest announcements, you stated that the latest financing, 21 million, would be sufficient to fund the first 6 months of the latest trial, and that exercise of warrants would be available to fund the last year of the trial. What is the likelihood that this exercise will happen at $5 per share? And is there a plan set out in the event that it is not?
Well, right now it doesn't look very good, does it? We are really heavily looking at getting organized and raising more money. The question is how, because at this market cap, it's very hard to actually raise money, but we have time until next year. And so we're moving forward with our drug. We take one step at a time, and we'll get there. I think that Churchill said, when you're going through hell, keep going. So we are going.
All right. Are there any upcoming catalysts that could drive a turnaround?
Well, I showed you the big milestones. A lot of things can happen in between, but a lot of it depends on what's going to happen, whether whether we get grants, whether we do deals, whether whatever. And so for the time being, the biggest catalysts really are that next year we will have the readout of the 6-month study. We will also know what the FDA says to the open label and what the FDA says to the Parkinson's, and we'll move from there.
All right. Now here we're arriving to our last prepared question before we open up live questions. How does the leadership team plan to restore confidence among investors who have endured significant losses?
Yes, I am really, really sorry about what happens now to our stock. We needed the money. We did a deal. It wasn't a very good deal, and we have been shorted a lot. Basically, as I said, we rely on our science. We do take one step at a time, and we'll make it through hell because our science is very solid. Our drug works. I showed you the forest plots that look very, very good. And I don't give up. We'll get there.
All right. Thank you, Maria. Now we will open the floor for live questions. So I see one hand up. There are a few more. I'm just going to start naming, and Maria will give you an answer. So let's start with Jim Stevens. I'm allowing you to open your mic there. You can activate your microphone. Jim? Hello, Jim. We're not hearing you. Okay, we'll try and get that worked out. Maybe you can fix your mic and we'll come back to you if you're able to activate it later. Let's go to Erica Breaking.
Hello, can you hear me?
Yes.
Yes.
So my question is about the synergistic, I guess the patents you got to use the GLP-1 drugs and the other one too, the ED drug. How is there a timeline for do those uses have to follow the timeline that we're already on for the approval of the drug? So 2 to 3, 4 years out when it's finally approved to use in early Alzheimer's. I'm following the Alzheimer's progress for the Alzheimer's.
No, we could actually start it yesterday. I mean, you have to write a protocol, you have to go to the FDA, which takes about 3 to 4 months. They have to give an answer. But no, we can start anytime if we had the money. And the reason is that Trulicity is on the market and is off patent, so we don't even have to ask anybody for permission. Viagra is on the market and is off patent, same thing. And our drug is in phase 3. So as long as we can show to the FDA that the combination is safe, we could do a large study.
Okay, thank you.
Thanks so much, Erica. Thanks for your question. Now we're going to Ramin Barnett. Go ahead.
Hello,
Ramin.
can you hear me?
Yes.
Hello to both of you. My name is Ramin and I'm from Manchester, UK. I've been an investor and a huge fan for the past 3 years. And as you can imagine, based on the current share price, I've been suffering quite a lot. My question is, despite all these positive advancement and good news, the stock price continues to plummet, and Maria, you perfectly explained why. Now, is there a possibility in the future that you may be left with no other option but a stock split, like reverse the stock split? And can you, I know is a $1 million question, but can you reassure your investors that good days are ahead of us and this trend is not going to continue till we get to the penny, basically, stock price? Thanks.
Yeah, well, there are no assurances. I'm amazed, totally amazed that the stock is where it is, given that our data really, really looks good. And, the question is, who is doing what with the stock? And investors like you and me, we don't have enough money to really push it one way or another. Whereas the ones that push it down, they have enough money to make money on the down. And so I can't give anybody any guarantees, but what I do know is that because we believe in the drug, we should all get together and actually buy stock. That's the only way out of it. Now, granted, if something happens on our side, if we have good news before next summer, that would help. And as I said, we are working very hard to get something going, but it's not as easy as that.
Well, thank you. I mean, I've done my share of encouraging my friends and family members to buy, and I've made some enemies so far, but I continue to encourage people to buy because I believe in what you're doing, and fingers crossed. Thanks.
Thank you. Thanks a lot.
Thank you, Ramin. All right, let's hear from Thomas Kamfeth.
Yes, can you hear me?
Yes.
Okay, yeah, thank you very much. And also thanks for the presentation and everything you're doing. I'm basically asking on behalf of my dad, who was enrolled in a Parkinson study from Germany. And my question is, will also patients from Germany benefit from the open label study?
No, I'm sorry, but we are not planning to actually have the open label in the EU. It's too complicated.
Is it possible for EU patients, since I'm a US citizen, to maybe enroll in one of the study sites in the US? Is that an option?
Yes, it's possible. Your dad would have to have residency here, which means he needs okay, what I'm saying is trying to help you. He needs an address. He can go home in between. He'd have to be here every 3 months.
Got it. I mean, I'm living here, so that from that end wouldn't be a problem to have an address. And I think I'm also living relatively close to one of your study sites, so that should be fine. Okay. Okay, great. Thank you and good luck and all the best.
Thank you.
Thank you, Thomas. All right, let's hear from Candice Galamini.
Hello, how are you? Can you hear me?
Yes.
Okay. I was just wondering if the study and the trial would be available for Parkinson's in Canada.
Well, we didn't have, the study was not conducted in Canada. So the open label would definitely not be there. And right now, we do not have any plans to do the next study in Canada. So I kind of don't think so.
Okay.
There's stock splits.
It's got
Fuck's sakes.
Well, it's got to propagate in the States, and if it works out, then maybe.
Mhm.
Oh, sorry, we're online.
Hi, Candice.
Oh,
Yeah, we can still hear you.
I'm
Oh, sorry.
Oh my gosh. That's my father from he's from Italy.
That's okay. You answered my question. If it's not available here, there's nothing I can do, right?
You can move. But in your case, you'd have to move. Open label, we can be a little bit more open-minded because it's not the official study.
A question I do have on that is her
Yeah.
husband. My neighbor is a doctor and his wife's a doctor. She had a second time now breast cancer, but there was a trial in the US, not in Canada, and our oncologist here in Vancouver got that trial to be started here through the company in the US. Is that an option maybe if we have a doctor here that wants to try to do the trial of yours of the Parkinson's? Can that work?
It is kind of an option. See, in cancer, you get a lot more special authorizations than in Alzheimer's. If something is deadly, a doctor can easily start his own study. In Alzheimer's, it would be more complicated. I'm not exactly sure how, but I do know that in diseases that kill you, it's much, much easier to get authorization to do something.
Yeah, she had a trial. she was given like just about 6 months, and now it's been a year and a half on this new trial, and she's really doing well, and apparently stopping the black spots or whatever that she has. But yeah, so I guess we'll find out if it's possible if it does a phase 3 on Parkinson's
Yeah.
happens. Okay, well, thank you so much.
Sure.
Bye-bye.
Thank you. And you're able to mute your microphone. Once your question's been answered, you can go ahead and click on that mic button to close it. Now we'll hear from Scott Sanchifer.
First of all, Brooke and Maria, thank you for spending this time with all of us. A close relation of mine is considering and would be a good candidate for your Alzheimer's trial. And one of the evaluations is whether this trial or another therapy would be better. So obviously I'm not going to ask you that. The question I have is in regards to obviously the funding component is, you know, through the 6 months. So if you were not to get the funding, what would occur in regards to the existing trial? It would stop, I assume.
Yes, it would stop, but I'll get the funding.
No, that's my next point. Maria, I want you to continue to fight for what's and the question is, from a funding perspective, you're looking for, obviously I can go and buy the stock on, I was just starting to look at how I could potentially invest in your company. And then the next comment would be is maybe Brooke, you could reach out to me and I could see how I could potentially find some investors that would be, you know, significant investors that would be also interested to support the fight.
Thank you. I really appreciate. Yes, we'll follow up.
Okay.
Thank you so
Thank
much.
Thank you very much. I really appreciate it.
Thank you.
Maybe one last thing. I was reading, why was there press in regards to your announcement on around April 29th that looking at the data, they consider it a failure, even though it seems like a significant improvement?
Because it's a subgroup.
Oh, it was a subgroup.
It did not work in the ITT population. It only worked in early Alzheimer patients.
I understand. That's helpful.
Okay.
But maybe one last question. Are you looking for a acquisition? I used to work in the pharmaceutical industry, so are you open to that as well, or that's difficult?
Well, you know, we are talking to interested parties, and it looks like they are more interested once we have the 6 months data.
Yeah.
Because that will confirm in a non-subgroup that the drug is a really, really good symptomatic drug. It's 3 times better than Aricept, which is by itself amazing. If you remember the graph I showed, with Kisunla and Leqembi not really improving cognition, if you think of combining it, you would have some disease-modifying effect by those drugs, and then the added symptomatic effect by our drug. If our drug on top of that shows disease-modifying efficacy, that would be fantastic, but I don't know that.
Right. Okay, thank you.
Thank you so much, Scott. We'll be in touch. All right. We can invite Jim Stevens to open up your mic. Your mic is open, Jim. Go ahead.
Hello, can you hear me, Maria?
Yes, we do.
Okay, thank you for all your hard work. I've followed your company for over 20 years. I'm part of the Stevens Capital Investments. Recently, with your share price under $2, we scooped a significant amount of shares. So we are willing to make a bigger commitment with you. We would like to see yourself and any of the directors, if they make open share purchases, we will try to match or buy more shares. We feel that you're on the right path. We see that your drug, which works as quick as 30 days, which is quite remarkable. And we see this could be a significant drug, in the future. I'm not sure why you not maybe partner with Biogen. Some large companies might want to just come in and scoop you up, you know.
Well, first, we are considering actually buying shares, but we are in a blackout period. We cannot until the end of March. So I hear what you're saying, and we'll definitely do that. In terms of pharma, okay, nobody has asked, so I'm going to bring it up because otherwise somebody's going to say I'm avoiding reality. We announced that we may have 200 million, and then the 200 million are not really here. So I was over optimistic, and I'm very, very sorry for that because I gave you all false hopes and expectations. Interestingly enough, I gave me false hopes and expectations because it would have saved all this thing that I'm telling you. It's dependent on FDA and money. So yes, pharma is interested, but they're slow. And it seems like once you are on a path of one antibody for one protein, the next step is one antisense, a molecule, RNA molecule, for one protein. And the next is one siRNA for one gene and protein. So to move from one protein at the time to 4 proteins together has been very hard to digest. So what's happening is that they look at us and they keep wanting more data to feel comfortable.
Oh, that's understandable. I mean, many times we invest in very small companies. For instance, we invested heavily in Insight Pharmaceuticals back in 09, 010. It was $3 a share. Once the investor realized the drug is there, the stock went from $3 to $150 a share.
Yeah.
Now we see significant moves in your stock. We believe in what you're doing. The drug will speak for itself at a point in time. But when you start making commitments after the blackout period, the investors will flood in. You'll say, I don't have enough shares to sell. So you stay what you're doing. You put some share purchases out there when you can. And myself and others will follow in. We're sitting on plenty of cash.
Okay, thank you. Yeah, we should talk how to best do that because I have to tell you, I think I'm really good at development, R&D, because that's my background, and I'm really learning the hard way how tough the market can be.
Well, that's a lot of times when you look at when there's opportunity, you scoop in when there's opportunity there.
Yeah.
I see that from other Parkinson's drugs that we've reviewed, you have the fastest effects in cognition and, you know, that development of stopping the dementia. Other drugs are taken 9 months, 15 months, 16 months, whatever, to even show some improvement. I mean, I can't believe that other large farmers aren't knocking on your door and say, let's make a deal, Maria.
I think they will, but we have to be a little bit more patient.
Yes. Okay. Thank you and good evening.
Thanks.
Thank you so much, Jim. All right, we have a lot more hands, Maria. Are you ready?
Yes, it's only 5:30.
All right, let's go with Michael Gopes. Your mic is available to open.
Great. Hello, Maria. How are you?
I'm fine, thank you.
Good. We've also been buying stock down here below $2 and believe in what you're doing. And thank you for continuing the fight. And I just wanted to make one comment on the deal you did, which raised important money. The average performance of a deal like that with those hedge fund type groups who buy and then sell all their stock and keep their warrants is about 30 to 50% decline in the month after the deal. So you're there. You're down 50%. So it's not unusual to see a stock go from $4 to $2 once you complete a deal like that. And if someone like Jim Stevens can complete a private placement with you, that would not have that effect. They would just purchase the shares, and it might if the news was announced, the stock would go up, not down. So I would definitely talk to people like Jim and try to put together a private placement where they just acquire a large amount of shares, and then you announce it and people look at it and go, okay, someone believes in you and it's a real investor, because those investors that participate in your deal are not really investors, they're hedge funds, and they're just.
Yeah, no, I understand.
Okay. My real question is, going into the FDA with your Parkinson's data, I know that the gait impaired group had a dramatic improvement, and I know it's a small group. Would it be possible the FDA would say, even though it's a small group, since they had such dramatic results, that they would offer it for compassionate use to those patients?
That's a very interesting question. I had not really thought about that. The issue is subgroups, usually the FDA wants you to confirm them, so we'd have to do another study anyway. But that's a very interesting question. I think when we have our FDA meeting, we'll bring that up. Definitely. Thank you.
Okay. And perhaps the Parkinson's patients who had the best results could be allowed force
Yes.
in
Yes.
that and make a lot of noise with the FDA, which would be great.
I agree. Yeah. Thank you.
Wonderful. And I guess submitting that data, would that encourage any major pharmas, or they're going to really wait for you to narrow down statistically significant trial results?
I wish I could give you a better answer. I stepped on my toes when I said we had the 200 million. I'm a little reluctant to really go into that. I don't know.
Got it. Okay. Thank you.
Thank you very much, Michael. Andrew, Ian, you are welcome to open up your mic. Andrew, Ian. Okay, if you're able to open it later, we'll get back to you. Donald Tice. Are you able to open?
Can you hear me?
Yes.
Okay, well, thank you, Maria and Brooke, for hosting this informational webinar. My question, I guess, goes to the NIH funding, the federal funding. You mentioned before that you do receive some of the funding from the federal government. And I was just wondering, do also your clinical trial sites receive funding? Is this going to be a big problem for you? Just trying to figure out how much you rely on the federal government to make this happen.
I don't.
Okay.
But I mean, it's nice to get free money. It's really hard to decide, or not decide, to understand exactly what's going to happen. Because on one hand, yes, there is some bloat. On the other hand, if you keep cutting, they're going to be slower. So the other thing is what you cut. It is hard. It may actually benefit us. And let me explain why. The FDA and the NIH have been very A-beta oriented. And somehow we got stuck in the A-beta train. And it has been very hard to get pharma or the NIH to look at anything that wasn't A-beta. So it may help us, but I really don't know.
Okay, thank you.
Thank you, Donald. Okay, Charles Overy.
Yeah, I think this is a follow-on to that question. It's probably pretty appropriate. You keep talking about talking to people at the FDA, and yet we hear in the news that there is no FDA left to talk to. Are they being responsive? Is this something that you actually see continuing business as usual, or is there a problem there? And as a follow-on to that question, we were expecting the Parkinson's open label to start in the end of this month, beginning of next month. And I think you briefly mentioned a couple of months you're expecting for an FDA response.
Yeah.
So maybe you could elaborate a little bit on what
that's
you're
not
seeing
the
at FDA.
FDA's fault. I don't want to
okay.
blame the FDA for that. That's our financial fault. Okay, let's blame who is responsible. And also the fact that we tried so hard to get the Alzheimer's going, and then the open label, we kind of pushed it a little bit out because, it's not as simple. I don't think that people really understand what an open label is. We keep getting questions from patients, why don't you just give me a bottle? An open label is pretty much a full-blown study. Patients come in, they get checked for everything, for all the comorbidities, for their whatever they have in the urine, whatever they have in the blood, whatever they have anywhere. They do all the tests again. All the tests are repeated every 3 months. They go home with 3 months supplies. From that point of view, it's a little less stringent. But an open label is a real study, and it costs $10 million a year. Okay, for 300 patients. Once we have 800, it's going to cost more. We still need to make the pills. We still need to bottle them. We still need to package them. And the drug is not that expensive anymore, but it's still way more expensive than aspirin. We think at some point it's going to be as cheap as aspirin, but give us another 3 years before we get there. So the FDA has nothing to do with the fact that we are late. The FDA to date has been exactly responding according to the timelines they say they have. And depending on what you ask, they have 30 days, they have 60 days, they have 3 months. And up till now, they have done exactly what they're supposed to do.
Okay, thank you.
It's
And thank you,
great news.
Charles.
Thank you.
Thank you so much. All right, let's hear from Robert Norsworthy. You're able to turn on your mic there, Robert.
Yes, can you hear me?
Yes.
Yes.
Yeah, I'm a Parkinson's patient, and I did a couple of studies. So I've been watching to see what yours is going to do, and I'm glad to see that you're using the biomarkers, the CSF biomarkers. There were no biomarkers involved in either study that I did, and I thought the UPDRS was pretty weak. And needed something much more quantifiable, better to, more precision. And so I'm wondering, what do you think you'll be getting from the CSF biomarker that you have in the protocol? And will it will the only biomarker I'm aware of is the Michael J. Fox, which is just useful for telling you if you have Parkinson's or not.
Yes.
It's not something that will tell you how it's progressing, measuring it at a finer grain.
Yes. Yeah, you're 100% correct. It's only a qualitative, but it's the only one. So if I want to know whether you have Parkinson's or not, and I don't rely on Hone and Yar and UPDRS, that's the test. Now, once we know in Alzheimer's, they have developed a lot of tests for a lot of biomarkers in plasma. Parkinson's is a little bit behind. So while right now we're going back and measuring everything we can in plasma of the Parkinson's patients in our previous study, so we're probably measuring your blood, there is less for Parkinson's. It's not as well developed. The assays are not as well characterized. They have much more variability. But, there is something in plasma. In CSF, Parkinson's has as good biomarkers as Alzheimer's does, almost as good. So if we take CSF, we're not just going to do the alpha-synuclein seeding assay to make sure a patient has Parkinson's, but we'll also do all the other biomarkers that we are trying to do in plasma and maybe are less predictive.
So is it something that you'll be doing more than one collection of CSF? And the CSF that I did, actually, I did a CSF just for PDMI though. But I'm wondering, is that something?
I don't know. We just had this discussion because obviously we don't want to stick patients if we don't have to.
Right.
So here is our rationale. When you come into the open label, we take CSF. We make sure you have Parkinson's.
Yeah.
Blood. And then you have a year. Okay,
Yeah.
here before we do anything again with you. No, you come in every 3 months to make sure you're living. But in terms of actually looking whether you have progressed or not, you have a year. And if the seeding assay is getting better or a new assay is being developed that shows progression in CSF, we'll take CSF. If it hasn't been developed, it doesn't make any sense.
Right. Okay, thank you so much.
Sure.
Thank you, Robert. All right, I think we have time for one more question here from Justin Higgins. Justin, you can open up your mic.
Hi, guys. Can you hear me okay?
Yes.
Thanks so much for taking the time. It's greatly appreciated. I'd just like to sort of reemphasize what some of the other probably more professional investors and retail investors were saying. We know a lot of people out here, and I think you should start taking advantage of that by reaching out to investors. I personally haven't sold a share of your company, and I'm buying like crazy down at this level because I think your stock is grossly undervalued at this point. My question is this. In discussions you've had with pharmaceutical companies where I actually have a lot of contacts, do they fully understand that the end point in P3 has been basically teed up to meet what they themselves do when they have failed P2 and P3 issues with their endpoints? They do the same thing that you have exactly done. Do they not know that you've taken that right out of their playbook? Are they not seeing that, Maria?
Oh, yes, they are. But remember, if it's my subgroup analysis, it's my subgroup analysis. If it's yours, I don't believe it.
Yeah. So it's just a matter of getting to that 6-month stage.
Yes.
Yeah.
And it may happen earlier. It really may happen earlier. Okay. But right now, I have a lot of people that are sitting on the fence.
I understand. So as a CEO, and I've ran companies myself, you had to make the decision that you couldn't risk running out of money. You had to pull the trigger to dilute, to get that money, to get you to 6 months, because if you don't get it at 6 months, it's not going to matter anyway. Is that really your mindset in dilution? I missed the beginning of the call. I apologize.
Yes, that is exactly my mindset. I mean, the drug works. I'm going to get it on the market by hook or by crook. And because where we thought we had the money didn't work out, we needed money.
Fantastic. Well, I look at it as probably one of the biggest buying opportunities out there right now. And we are buyers and we will continue to buy aggressively. Unfortunately, I would love to see it above 5, because if we could get it above 5, we could really do some buying. But unfortunately, there are some rules around that. But thank you for taking my question, and good luck to everybody. I appreciate it.
Well, thank you so much. Thank you. By the way, I want to thank everybody. You were so kind. You really built me back up, because it has been really, really tough lately. It seems like, I want to do it so badly, and I don't want to let patients down. And I do want to do the open label. And the question is, where the hell do I get the money from? But we'll get it.
Thank you. You've got a lot of positive feedback here, Maria. Well, our time is up for now. So like Maria said, thank you all for your very insightful questions and encouragement, and for participating in today's webinar. Of course, there are a lot of questions that could not get answered. So here in the chat, we're posting the email that you can reach out with more comments or questions. Feel free to get in touch with Maria and her great team. And a special thank you to Dr. Maria Maccecchini for addressing many important questions coming from the patients community. And yes, to answer many of your questions, yes, today's webcast will be available on our website shortly. So for now, thank you to all for joining, and we'll say have a great day for now.
Yes, thank you very much. Have a wonderful evening.